My name is Noel-Marie Plonski, and I'm a graduate student in Dr. Piankisko's lab at Kent State University. I am currently using bioinformatic approaches and tools to study viral host interactions. The title of my presentation is RNA Editing Landscapes, a new model for biomarker discovery in neurological disease. Although there are many types of RNA editing, we focus on adenosine deaminase acting on RNA ADAR editing. ADAR deanimates adenosine to inosine, which results in an A to G transition. There are three ADAR loci, ADAR 1, 2, and 3. These ADARs are expressed and edited in many tissues, including the brain, and play an important role in many developmental processes. It has been shown that activation of the TLR3 pathway by viral infections leads to increased transcription of a number of genes, one of which is an isoform of ADAR1, ADAR1P150. This means viral infections can lead to changes in ADAR editing footprints, not only in the virus, as well as potential having unintended consequences in the human transcriptome. For a biological example of ADAR editing, we look at one of the most commonly studied editing sites in the GRA2 receptor, the QR site, where this site needs to be edited at nearly 100% to stop calcium signaling during development. If this channel is left in the unedited isoform, then it can create an exotoxic condition in the cell, leading to cell death. Another commonly studied gene with known editing sites is serotonin receptor 2C. There are 5 editing sites in different patterns that create 24 known differentially functioning isoforms. These editing sites have been shown to affect both serotonin signaling and overexcitability of the neuron. Serotonin signaling has been implicated in mood disorders and suicide since the 1970s. However, these are only 7 of the 152 known ADAR editing sites located in 98 genes that make up the excitome. A gene enrichment analysis of these genes shows the excitome genes are involved in pathways responsible for cell signaling, ion homeostasis, metabolic function, and other transcriptional and translational processes. This leads us to our central hypothesis that others have also shared in recent publications. Maternal immune activation, postnatal infection, chronic inflammation, and stress in the lifetime of viral infections can alter normal spatial temporal regulated ADAR editing patterns in the brain, which can then ultimately result in a spectrum of neurological complications, depending on the time and location of the changes to the ADAR editing landscapes. These neurological symptoms include symptoms as obvious as microcephaly, but as silent as mood regulation. The first step was to create a fully automated pipeline using a new mathematical model to map ADAR editing profiles. Our pipeline aid utilizes standard open source tools for genomic alignment, transcriptome assembly, variant calling, and variant prediction, combined with our custom EX toolset to create the editing landscapes. The new model includes item response theory, utilizing the Guttman scale, and machine learning theory random forest analysis to score, rank, and order editing sites in any condition. To test one of the parts of our central hypothesis that changes to ADAR editing landscapes can be used to predict biomarkers for depression or suicide, we use publicly available datasets. The datasets together consist of 324 samples. Each sample was assigned a score by Guttman scale based on editing sites. The higher the Guttman score, the more editing is present in the excitome. Here we see significant differences between suicide and natural causes of death, biological sexes, and between all the brain regions. Here, and for the rest of the presentation, natural causes are shown in blue, and suicide is shown in red. Here, females are on the top panel, males are on the bottom, and the regions are shown from left to right. The highest editing is seen in the prefrontal cortex. In females, editing is higher in suicide in the cingulate gyrus, and lower in suicide in the prefrontal cortex. Females also show more editing in general and more significant differences in editing amounts. When we look at Guttman rank order, we set 3 categories. Category 1, completely edited in every sample. Category 2, edited in only some samples. And Category 3, not edited in any sample. We set the Guttman order for the female anterior insula as a reference. Here the females are on the left and the males are on the right, and the regions are shown from top to bottom. We see here the natural causes of death. The editing patterns are pretty similar in females with only a few category switches. But when we compare suicide to natural causes, we see a significant number of changes both in category and down. Males, on the other hand, show more differences between regions and cause of death, suggesting major differences between males and females. This suggesting less of a role of editing in suicide in males than females. Using random forest models, we create a model with an 18% error rate using excitome editing frequencies. These graphs here show the top editing sites using prediction of suicide or natural causes of death. If we look at the top row, which are the top predicted biomarkers, we see clear clustering of suicide in red and natural causes in blue. If we take a closer look at the significant differences for some of the top genes, we see changes to the serotonin receptor, but only in the nucleus accumbens. All 5 editing sites show differential editing, and they appear to be different in males and females. Females, again, showing more overall editing. This suggests serotonergic tone and dopaminergic reward regions of the brain may be an underlying biological mechanism of suicide. We also see significant differences in calcium channels, including the glutamate channels GRA2 and GRIC2 at multiple sites. Here again, we see differences between males and females, as well as brain regions and suicides. These would suggest a role for glutaminergic signaling in multiple regions underpinning suicide. The top-ranked ACP1, which is a gene involved in cholesterol metabolism, shows significant differences in all brain regions and is pretty similar in editing patterns between males and females, and in most regions showing less editing in suicide. Moreover, this gene is also expressed in edited blood samples, so it can be used as a clinically relevant biomarker. The last gene I will mention from the biomarker list is IGF-BP7, which is an insulin growth factor binding protein, and has been shown to be more susceptible to proteolytic degradation with decreases in editing, like that seen in the subveliculum in prefrontal cortex of females and cingulate gyrus of males. In summary, ADAR editing profiles of the excitome can be used to not only find biomarkers to predict suicidal risk, but also provide insight into the biological mechanisms, including alterations of calcium channel signaling and plasticity, and dopaminergic reward pathways through serotonergic signaling changes. We have the ultimate goal, finding a better diagnostic and prognostic tool for mental health care professionals in treatment of mood disorders. For example, assessing an individual's response to drug therapy based on editing profiles or risk for suicidal or homicidal behavior. However, much more investigation and more data needs to be analyzed before any of these things can be achieved. With that, I'd like to thank the organizers for inviting me to talk, and everyone at the P&Q Skill Lab, and my other mentors at Kent State, other collaborators shown here.